Okay, hello everyone. Today I'm going to be talking about pseudotime analysis for snapshot single cell RNA-seq. So like snapshot meaning like you've done one experiment, it's not like a time series experiment. Um, so first of all, I think I want to cover like what is pseudotime, uh, which is a big question, I think. This is a figure I took from a review about pseudotime analysis. And I think that first of all, there's kind of like in terms of nomenclature, some people say it's pseudotime, some people say trajectory inference analysis. And they're kind of like you can think of them interchangeably, and we'll go over like why is it even called pseudotime in the first place. But at the top here, you can kind of see like this one is actually showing a cartoon of like if you did have a time series data set, where you would expect that like cells from the day one time point would turn into the day three and then day five and so on. How do you kind of like stitch them together in that ordering when you've collected all your data? And they broadly break it into two different types of models, where first you start with your high dimensional gene expression matrix, so like a cells by genes matrix. And in this one, you're kind of reducing the dimensions to get like either like a PCA or a UMAP or whatever dimensionality reduction. And then you're applying some kind of graph learning into this space. And then the graph is kind of like the backbone of the trajectory. And then you apply some other algorithm that will kind of take the cells, and it'll take the graph, and then it'll allow you to assign a pseudotime or trajectory like time point to each cell. And then the other type of analysis that they talk about in this review paper is probabilistic models. So this is kind of similar, but it's kind of based on instead of this dimensionality reductions, it's kind of based on like linking clusters to each other, like which cluster, like if you have 10 clusters, is cluster one like likely to give rise to cluster two or cluster three and so on. So there's like probability of transition probabilities essentially between each cluster, and then you can use that to extrapolate pseudotime and trajectories.
So for the second strategy, you're only getting discrete pseudotime bins. You're not getting like continuous pseudotime bins.
They at the end of it, it gets you to the continuous, but it's kind of based on clustering rather than based on dimensionality reduction. And like I think I'll go into these later, but to like this top approach is like monocle 3, and this bottom approach is like Paga, if you've heard of those. So I kind of want to talk about like a couple of the questions we might have about like what about pseudotime in general. So like I think typically like the figure on the right is from my research, and it's like a very typical like what you would see when you do single cell RNA-seq. So we have gene expression data and we have clusters, right? So these are like cells grouped into discrete bins. We usually get these by running something like Louvain or Leiden, which are called like community detection algorithms. And yeah, so like I said, this kind of places cells into discrete groups, which we usually refer to as clusters. And then the alternative is trajectory inference or pseudotime, which places cells on a continuum rather than into discrete groups, which could be helpful. So some burning questions we have is like, okay, when should I use clustering versus pseudotime? And are these approaches complementary? Are there cases where we should not use pseudotime? And what are the tools that we should be using for pseudotime analysis? And sorry if I'm like interchangeably calling it pseudotime or trajectory, but I think that I'll explain why it's called pseudotime like right now. But this is the original paper that introduced it back in 2014 in Nature Biotechnology. They were trying to study development of like muscle cells. So that is a temporal process where like you have a progenitor, and then time goes on, and then you have a mature cell. So they did have time series data, or I guess like three different groups: the proliferating cells, the differentiating cells, and then the mesenchymal cells. And they kind of wanted to take that data and draw like a path through it. So they didn't have a word for like trajectory inference or pseudotime. They came up with the word pseudotime, right? Even in the title of the paper. And this is on the left side. This is kind of the overview of how they did it. So they first have like gene expression data, single cell, and then they have a dimensionality reduction, and then they applied an algorithm called a minimum spanning tree. that basically connects the cells into a graph, and then they can order the cells based on like assigning a start point. And then they can just do downstream analysis like differential gene expression and stuff like that. So the minimum spanning tree is shown in panel B for this data set. And this kind of looks like, I think like in comparison to like this kind of plot we would see nowadays, this is like, 2014. So this is like kind of like looking at like a cave painting or something compared to like the Mona Lisa. So there's still some, this is like our ancestors and we can kind of see where they're coming from, but like the algorithms have improved a lot since 2014. But I think this is also a good point to notice that like where are like the trends in the field, because I think after like around this time of the early days of single cell, a lot of the algorithmic people were actually working on the trajectory tools. Not as much these days. A lot of it these days is like integration and like spatial omics and stuff. But these trajectory tools are still useful, of course. The same group followed up their monocle algorithm with something called monocle 2. And instead of a minimum spanning tree, they're using something called reverse graph embedding. So it's just a different graph method, basically, instead of a minimum spanning tree. But it's a very similar overall. You have to have single cell data. You have to have a dimensionality reduction. You're building some graph, and then you're kind of assigning the pseudotime based on the graph. And in for monocle, it is like required for all three versions of monocle. You have to kind of manually assign like where is the start. So you have to use your kind of like a priori knowledge. And yeah, this one came out in 2017 in a Nature Methods paper, which is like, so they were actually talking about like the method in this paper. Their follow-up to that was in Nature, but it was actually like about this, the paper was about organogenesis, and they just like threw in a new method. They're like, here's monocle 3. So it's like actually, unfortunately, not like super well described. But similarly, they have like another, like, it's like each one of their methods, they have like, let's use a different dimensionality reduction, let's use like a different graph. And they didn't do any quantitative like benchmarking to say like, why would I pick one or the other. But in this paper, they just like generated a ton of data and they did a ton of different trajectory analysis. And yeah, they typically use like UMAP, and then rather than like monocle 2, would just kind of make its own dimensional reduction. Now, if you just have a UMAP or a t-SNE, you can just make do a pseudotime on that directly. And this one has a very nice documentation website, and I've had like good experiences using monocle 3. So I think that's like the one I would reach for first in my toolkit if I was doing like an R-based analysis. But yeah, now I kind of want to talk about like what kind of work has been done to benchmark and like how would I pick out of all these different tools. This was a paper in Nature Biotech from 2019 where they have a bunch of different data sets and they had 45 trajectory inference methods, and they had a bunch of different metrics, and they tried to like grade, like give a report card to each of these pseudotime methods. And they tell you they have some idea of like, oh, we know like in their data sets they have an expectation of like which cells should be connected to which ones, and like they're kind of grading them based on the accuracy and how well does it scale to larger data sets, and as well as like usability of the package. Like, is the package documented? Is it maintained? Is it hard to install? They account for all these things. And they also in this paper, they developed like a pipeline called Dyno, DYNO, that is like a unified input to 45. I think they expanded it to 55 different trajectory inference methods. So that's pretty cool. It includes like inputs, and it has like plotting that can be used, like downstream plotting that can be used for any of the 55 methods they have. That being said, I actually haven't used Dyno. It just looks nice. I haven't used it though. But yeah, basically they're looking at these different things like topology and like where are the branch assignments. So this is like relating to whether there's like, are there like cycles in the trajectory? Is it a bifurcation or a trifurcation, or is it linear? So on and so forth. It's looking at like distances that these metrics are computing between cells. And it's finally looking at like, how does it like rank genes along the trajectories? So this is kind of like one of the main figures of this benchmarking paper, where it's scoring different like each row in this table is going to be a different method, and then it's going to tell you different information about it. So which kind of topology is going to handle, and then what is the like how good did it do on the different benchmarks, the different tests, like for accuracy, scalability, etc. One of the ones that scores pretty well on like most things ends up being something called PAGA, and I'll talk a little bit about that later.
What does the like color mean here?
Their color scheme is kind of weird where it gets lighter the higher it is.
Okay.
Darker is
They're
worse.
using two things to represent the same variable.
Yeah, it's
Got
the like length of it is like yeah, so this one's like light colors and it's big bar. So that's good. The dark color is bad. I don't know why they did that. Yeah, so they did evaluate a lot of different things, and this can kind of tell you like oh, maybe if I have like see how like the cycle here is dark, but it's not down here. So like these ones, like if you knew you had like like if you're trying to study something that you knew was cycling, you would immediately be like, oh, I can't use any of these ones down here. I have to pick one of these. And then on the other hand, like if you knew you had like a bifurcation, you might want to pick a tool that is specialized for that. Like Paga is more general for any topology, so maybe you would pick something that has is specialized for that. Yeah, some of the algorithms have like prior knowledge about like the type of like splits that they're expecting, like whether it's a bifurcation or a trifurcation or a cycle or something. Also consider whether you want to run it in R or Python, right? The next one they're talking about is like similar, but I guess more detailed. I think the one that's like really interesting is scalability, where they looked at like 1 million cells times 100 genes, or like 100K cells times 1,000 genes. And some of these take like prohibitively long. Like it looks like days for some of these. Whereas, I mean, I think most people would probably have like 100,000 cells or fewer. So like Pago seems to be looking pretty good still, where you have like 7 minutes instead of like a day. Monocle looks like what, 26 minutes? Uh, this is like also to be clear, the Monocle ICA, I believe, is Monocle 1. Monocle DDR tree is Monocle 2. And Monocle 3 was not in this because it was like published around the same time. And yeah, obviously this is 2019, so there's been more methods since then, but still this is, I think like I was saying earlier, the algorithmics people have kind of moved away from this. So like this still I think is covering a lot of the methods that are out there. So it's still helpful. Yeah. Also like I think this is more qualitative, but they're like quality of software in the paper. Like the bigger the dot is, the better. So they said like Paga's code assurance was low, but I'm not quite sure what that means. But
is that just like tests?
Oh, probably. Yeah. I think that back then, like that was true maybe because I think that the ScanPy, like Paga is part of ScanPy, and I think that package has come a long way in terms of the documentation stuff. Like in 2019, 2018, it was like brand new, and it was like I think it was like one guy working on it. So it's definitely like I think nowadays this would probably be I think it would be like Paga would be good for all of these. The paper is really short though, but there is a paper. And you have a question?
Yeah, how come there are metrics even measured for like the methods that said they couldn't even like assess certain trajectories? Like, weren't there a whole bunch of them that couldn't handle cycles, but everything has a dot here?
Where is it for a
Like, if you go
cycle?
back,
Oh yeah, that's a good point.
there's a whole bunch of like nothing in the
Yeah.
three methods are supposed to be able to handle cycles, yet like some of them are outperforming like the methods that said they were able to handle cycles.
Oh, that's a good question.
That's something the reviewers
Accuracy.
should have asked.
Maybe they say in the paper, but I don't know. Their last figure in this paper is literally like this is the flow chart for like how should you pick a pseudotime method, which is like really helpful. So like, do you expect multiple disconnected trajectories? So that's like, let's say like I'm thinking about the brain. So like if you have like microglia and astrocytes and oligodendrocytes, there are 3 different cell types that each could have their own trajectory. So like, yeah, okay, I do want that. So then it tells you which tools are applicable. So it's like pretty much just Paga, I guess. Or of course you could just subset your data and then that question, I guess, no longer, or you have a different answer to that question. But yeah, I think still like the takeaway seemed from this that Pago was an overall good trajectory analysis tool for various reasons. But yeah, they do have this like bottom left, like this suggestion that they should you should confirm your expectations with like using a method with a free topology, which is like Paga, I guess, and you should try at least two to see like if they give you similar results.
What is free and fixed topology?
The free would be like the fixed topology would be like linear cycle bifurcation, and then the free topology would be like it allows any connections in the graph. Yeah, they do have this nice website you can go to, which has it's actually like a collection of I think 4 packages that like Dino is the one that you can run the different methods, and they also have like Dyn plot and Dyn eval and Dyn methods that are like if you had a new method and you wanted to add it and stuff. Or if you wanted to like, yeah, I think you could, if you had a new method, you could run their benchmarks on it to see how good it performs. But yeah, ultimately this is like a 4-year-old paper and it doesn't look like this package has been updated that in 4 years, but I think it still should work theoretically. Yeah, so Paga is like the one I kept talking about that was like pretty high scoring in this. So this is a paper from the Tyus lab from 2019, which is like the group behind ScanPy. It's a pretty simple approach where they just have like you have your gene expression data and you have done like clustering on it, and then it's basically doing it computes like similarity between clusters based on their expression level and can kind of like draw an edge between them based on that. And then they can take that graph and they apply like some kind of like diffusion method to compute the pseudotime based on what you call the root. So overall, it's like a really fast algorithm, and it's like built into a tool that you already use, like a lot of people already use. So that's a good one for sure. I think like as an aside, part of this conversation, I wanted to talk about RNA velocity. This could definitely be its own whole topic, but this is like a pretty like distinctive set of trajectory inference or analysis tools. And it's kind of all based on this idea that when you do single cell, you have a mixture of unspliced and spliced RNAs. And then you can kind of get an idea of like not just like the quantity of RNA, but like the velocity. So like, is this gene increasing or decreasing with expression? And then that's like, in this paper, they extrapolate that to like a vector field where like the arrow is pointing to like a cell that is more similar based on these changes in gene expression in the velocity. So this is like one of the figures from this paper. And I think like basically in panel A, they have like these neural progenitors, like in yellow here, and then they have like a bunch of distinct cell fates. So like granule cells, astrocytes, radial glia, and then these other types of neurons. And then yeah, I'm just gonna like skip over to panel C where they show this vector field laid on top of the t-SNE plot, where like the arrows are kind of pointing to like I'm this cell and like based on my velocity, I'm like going to turn into that cell. It's a nice idea and it's pretty visually compelling, but they actually don't really like directly have like in this paper, they don't have like a pseudo-time calculation here. There's just like this vector field. So like how can you do this like you can't plot like gene expression versus pseudo-time because that's not one of the outputs. But then this group followed it up. This is the same group that made Paga using something called they called it SC Velo. It's pretty much a re-implementation because this was a R package called Velocito. This is a Python package called SC Velo. Mostly just a re-implementation, but to make it novel, they're like, oh, what if we like slightly changed it? So they have this like steady state model is the original, and then they have a slightly like they change these equations a little bit. So there's like a dynamical model in their ODE. So yeah, but overall, like I think the results are quite similar. Unfortunately, with these RNA velocity papers, they really don't do any benchmarking. I'm not aware that any has like been done. Maybe there has, but I'm not aware.
Can I ask something?
Yeah.
I don't understand much about this context, but basically, the difference between RNA velocity and pseudotime analysis is that in pseudotime, you're looking at some sort of a developmental process.
You don't have, it doesn't have to be developmental process, but I'll go into that, but it doesn't like have to be.
Okay.
It can be like more abstract.
So you're just looking at how the gene expression profiles are changing and you're grouping cells that have like a similar.
Yeah, it could be like, you can think about it like also like, if you have like a cell that's like been like, if you stimulate it, it'll like we're using a drug or something, it'll probably change in response. So you can use pseudotime to model the change, but that's not necessarily development. And then it might have like an activated and a relaxed state. So that's like, it will go back and like, it could like oscillate. Yeah.
Also, another thing is that in that case, do you have to make sure you know all of the variables that can affect that gene expression? Because you can see like, okay, these genes, their expression are changing in a similar direction, but what could, is it because another cluster, like another set of genes or RNAs are affecting them? Or is it the effect of this other like drug you said? Like, I guess I just feel like you can see a change, but how do you know what to relate it to?
Yeah, I think that's a question for like the downstream interpretation, because I think that none of these really account for that essentially. But like this method doesn't have like, this original one doesn't have like a pseudotime calculation. I didn't show it in this figure, but they do have like a way to extrapolate from the velocity to pseudotime in scVelo. So that's like a nice addition to it. But like I think us like a lot of the older ones, like the ones I was showing first, they are semi-supervised, so you tell them where the start is or where the end is. This one is like, oh, the directionality comes from the arrows. And I know like, unfortunately, just like the landscape of publication lends it to like, you hardly ever, even when you're like a methods person, you hardly ever get to show like the limitations, like when does this fail? Because they have to show like, when is this best? So like, this is like the best case scenario where it like looks good and it makes sense. It goes from the progenitor to the like more mature cell. But I think like, especially with the stimulation stuff, I've heard people at UCI and other places that have like, they run the RNA velocity and the arrows are like going like all over the place or like backwards from what they would expect. So it's like, then it's like unusable essentially. And like, you can also like, it ultimately relies on like these splice and unsplice quantities, which can vary a lot depending on like the sequencing batch. So like, and there's not any way to correct like batch correct for RNA velocity. So pretty much all these RNA velocity methods they show you are like n equals 1 single cell sample. They don't actually, like, you can run them mixed, but I've not really seen that being shown in the paper. Other people like using them will do that, but then it's like the results can get like messy. And like for that reason, I've like run these programs a lot, but I've actually never decided to put them into any of my papers or anything, because I'm like, it's just confusing. Like I should just use the method where I can like pick the start because I know what the start is already. Yeah, and people are still working on new RNA velocity methods. Like this is one that came out last year, and it's trying to use like for single cell multi-ohm data, they're trying to use chromatin accessibility information as a new parameter in their model. So like before it was just looking at trying to model transcription, splicing, and degradation. So this actually has two new equations that are for chromatin opening and chromatin closing. And they claim, like, I think that from a theoreticalist perspective, you're like, okay, you're like modeling more of the biology. That's great. But they're like, this is like what they show to compare basically is like, here's some data set and here's our method and then here's scVelo and like look how much better like our arrows are. And I'm like, but I'm like, I don't, yeah, I'm like, where's the real benchmarking? I don't see it here. But yeah, this is what I was meaning when they say latent time, but that's kind of like RNA velocity extrapolated into pseudotime. So it's like a continuous 0 to 1 pseudotime kind of value. Yeah, so that's like that's all I have for like the discussion of different types of methods, and I wanted to show a couple of applications. Am I okay on time? I have a
Yeah,
watch.
sure.
Okay. So I'll show a couple of examples, and they'll be like, I think a couple of them are from my research, so I can explain it better. But this one is like a famous one in my field that is like, it was an early single cell study. So this was 2017, and they were studying Alzheimer's disease using a mouse model. They have like wild type mice, and they have the mouse model, and they did single cell RNA-seq of the immune cells. So they have the immune cells of the brain. So mostly microglia, but as well as like regular like T cells, B cells, monocytes, and so on. And using pseudotime analysis, they found a new type or like previously uncharacterized type of cell.
But I have to leave earlier today, so when you guys leave, can you just make sure this door and this door are open?
Yeah, perfect. Thank you. Okay, anyways, they were able to find this like disease associated microglia cell type using pseudotime analysis.
Wait, so this is a mouse,
This is in mouse.
and they showed their graphical abstract of human brains.
Yeah, and they like usually say, yeah,
Okay, just.
that is a human brain, but it's definitely a mouse. That's why it's in cell. They're like,
in human. Okay, I'm nitpicking.
so yeah, this is like one of their main figures where they have like this is their microglia population. And it kind of looks like there's two groups here. And they're looking like this is just like the middle plots are like expression of different genes. And then like they did apply a pseudotime that kind of goes from like the left to the right. And then on the left side, you can see like these histograms showing like fraction of cells at these different points along pseudotime from like 1 month, 3 months, 6 months, 8 months. And this is so like for Alzheimer's, this is kind of like progression of the disease. As you get older, the disease gets worse, right? So there's kind of like the younger mouse is like healthier, and like that's where these microglia are on the left side, whereas the old mouse, all the microglia are on the right side. And then they can do these like differential expression tests to see like which genes are there that are more expressed on the left side versus on the right side. And they found like a bunch of these different genes like TREM2, TYROBP, and so on that are like expressed in the disease-associated microglia. A lot of these actually happen to be like genes that are previously associated with Alzheimer's through GWAS. So that was kind of like a big finding for them. And then like my own research, we kind of like wanted to do something similar, but we looked at it with human data, and we had a TAC-seq and RNA-seq data together. And like I think we use Monocle 3 for this, but it wasn't like explicitly like they didn't explicitly tell you like you could do this on like integrated multi-omic data, but we're like, we should just try and see what happens. So the UMAP we have on the top left is like an integrated object of a TAC-seq and RNA-seq. And then these heat maps that you can see in panel D are similar to the ones that they showed in this panel F, showing like the genes that are differentially expressed along the trajectory. But we also showed like chromatin accessibility changing as well, since we have the TAC-seq data. We also looked at kind of similar to their plot here. We showed that the proportion of nuclei from Alzheimer's brains was increasing along the trajectory. And we also looked at these like gene modules. So these are actually the genes from this paper that they identified as like stage 1 and stage 2 DAM genes. And we were able to basically see that like one of the stages was like not really there in the human data at all. It was like only it was pretty much like a mouse exclusive versus human exclusive. Another thing that we did was actually like instead of just embedding the cells and ordering them, we actually embedded the genes as well using a different approach. So we had like an ordering of cells and an ordering of genes, which like kind of helped us to characterize it in a more like complete way, I guess. Another thing that I did in one of my projects was using single cell long read RNA-seq and then doing like co-expression network analysis and then seeing how does those networks change along developmental time. So I used this data set that was published in Nature Communications of the hippocampus in a 7-day-old mouse. And then I took this radial glia lineage. So it's like the radial glia are progenitors that give rise to astrocytes or ependymal cells, or these other neural progenitors that will give rise later to neurons. So I just took that subset of the data, and then I computed 3 different trajectories using Monocle 3. So the way I did like that to get 3 different trajectories was like subset. I know that it always starts with the radial glia, and there's 3 different fates. So I just like subset it to be like radial glia and ependymal, radial glia and astrocyte, radial glia and NPC, and then just run Monocle 3 different times. And then I have 3 trajectories. After that, I used my package called HDWGCNA to find co-expression networks in this data. And then I wanted to see how the expression of those networks changes along the 3 different trajectories. So on the plot on the left, we can see that some of these networks are like exclusive to different cell fates, like this red, so yeah, do I even have it? Yeah. The red line is the ependymal trajectory, and the blue is the astrocyte, and the orange is the NPC. So like these 2 modules up here are expressed in the transition from radial glia to ependymal cells. Whereas like this one down here is like, or interestingly, like actually like astrocytes are transcriptomically similar to radial glia, even though they're like morphologically distinct. So some of these are kind of like, they're there and they just like go down, but they're not really expressed in the adult or like the more mature cells. And then there's also like similar dynamic patterns for like modules that are associated with like the neurons. I think I have a couple more slides. This is kind of an example of maybe like a pseudotime analysis that like I just didn't really understand, or maybe it confused me. This was a paper of like that was in Nature, where they're looking at human multiple sclerosis samples. And this is just one of their main figures, that they were looking at this excitatory neuron population that they like termed layer 2, layer 3. So they're coming from the upper cortical layers, basically. And then they then just took that cell type and ran a pseudotime analysis on it. So you see like this is just like a blob, basically. T-SNE or UMAP doesn't say. I mean, it probably does in the text, but it didn't say in the figure. And then they use Monocle 3, which is the same approach that I use here. But then like it's hard to like really interpret this. They started like they anchored the graph at like at the start as where the like controls were the highest, right? And yeah, they have 3 different conditions. So like the data set the data they took from like were from acute or chronic lesions in the brain from the MS. So they were kind of hoping to see like this trajectory that goes from like the control to the acute to the chronic, and like how are the neurons changing along that axis. But this might have just been better to do like regular differential expression to like compare those groups, like control versus acute, control versus chronic, acute versus chronic.
Isn't also the CUX2 expression is changing from control to MS, right? Is that
but I can't see it, you know what I mean?
Yeah.
But this is in Nature, so whatever. So like I think that this is maybe like an example of like they should have done something else, or they could have like it's not convincing me that this is like a good use of pseudotime, I think. Especially because like I think like it's also like overall it's like an abstract concept kind of, where like I think it makes a lot of sense when you say it in the context of development, like this is turning to that. But then it's like, okay, we know that mature neurons are just like they're there your whole life pretty much, right? Like they're not turning into something, they're not going anywhere. Like they might change their expression program so that you can model it in that way. Like how is it like the neurons are changing in disease? So like what is the path that the transcriptome takes? And I think that's what they're trying to convince you of. But ultimately it might just be like simpler to and more compelling to just have done regular differential expression.
Also, this makes people say like, okay, so what? Like I just don't get.
Yeah, like especially, yeah.
What is great?
Yeah, I'm like, I don't understand this plot on the side either. Like, what is?
It's
It's like a bar plot without the bar, or the box and whisker plot without the box.
had it like, they could have had it as like your line plots that you had in the previous picture. Like,
Some quantification, something.
Yeah, but like the way that they did it is like it's kind of taking the 2D picture of the UMAP and compressing it into 1D. That's just the pseudotime, right? But then it's
but
like
then it's 2D again.
they've lost their information.
Yeah, so it could have just been a KDE.
Also, it's throwing me off at the number of L2, L3 units going from 1500 to 0.
Oh yeah,
What?
yeah. But
This paper is a good one to nitpick if you want to read.
their
the whole
data's
thing.
good. I have this data that I downloaded, so but yeah, I think that like, well, I don't know, like it's easier to say this in retrospect, but maybe at the time this made sense. But like yeah, so that's like kind of all that I had to show. Like there's a lot in this space, but there's like a lot of different tools, and like you kind of should think about like whether it makes sense to even do pseudotime. Like, does it actually provide you more information than just clustering? Arguably, in this paper, they looks like they have two clusters, and they could have just done differential expression, and they would have probably had the same conclusion.
But their ordering for the gene expression profiles like ends up looking very interesting.
True, and like yeah, the ordering is nice, and like you can see like I think I don't have any examples, but sometimes there's like a critical, like it looks like there might be this critical population. Like what is the what are the genes that are expressed in like in the transition state? But they didn't really focus on that. So that would be something that you could do like with pseudotime you might not be able to do with clustering. But yeah, this is just the same exact as like my opening where it's like, what is pseudotime and like how is it different than clustering? I hopefully covered a lot of these things, but this is like, yeah, this is a pretty big topic, and ultimately I think it's like complementary to clustering, but not necessary in many cases. If you have a developmental data set, you should definitely do it. But even then, it's like the advice from the Dino paper that you should try more than one method, I think holds true, because like they can just be giving you like, especially with RNA velocity, they can be giving you like weird results that are hard to interpret. So trying to like reproduce it with an alternate method is smart. There's also a lot of like downstream like once you have the pseudotime, there's like, you know, downstream things you can do, like identify, like basically doing a differential expression test where your variable isn't like group 1 versus group 2, your variable is the continuous pseudotime, right? Monocle 3 has a module to do that built in. So like overall, I think like Monocle 3 is a pretty good like end to end, like it does everything I need it to do. And yeah.
It's a random question. Do you know if these are also usable for like epigenomic marks? Like if there's like a
Yeah, definitely.
stuff.
So Nagin's asking if we can use pseudotime for like for epigenomic data, and you definitely can. So it just kind of has to be like, I think the input to Monocle 3 is pretty much just it's gene expression data, but it computes a UMAP based on it. So you can like give it the any UMAP or any dimensional reduction, like if you have it for methylation data or a chromatin accessibility data. The same group that made Monocle had made a tool for attack-seq data analysis, so they have a tutorial for that already, like for doing the trajectory analysis on attack-seq data. But yeah, I think like maybe it's like not, I think for most people it's not useful, but it certainly is flashy. Okay, you have a question?
Yeah, so back when you were talking about the benchmarking paper, you brought up the capability of some pseudotime methods to analyze disconnected lineages. Why would you do that instead of just doing a re-embedding of your known lineages that, or like your connected things that
if you're like lazy, I guess. Like I think Monocle can literally just like it can run the like where's an example? So okay, this is a UMAP on the left, right? And like there's different clusters, but in the UMAP space, there's like a couple of blobs. Like this is one blob, this is another blob, this is another one, another one. And monocle, like it does clustering, and then it also like so it gives you a cluster ID, but it also gives you something called they call it a partition, which is like it literally just corresponds to like separated blob in the UMAP. And so by default, monocle 3 will like run the trajectory separately for each partition. You can force it to put them together, but I think that is just like a recipe for disaster. Because then you'll have like cell types that you know aren't turning like they're not giving rise to each other that are like connected in the pseudotime. So yeah, I think that's another thing I can say is like this is like user like prone to user error. Maybe more than clustering is. What's your question?
Think about it.
You should think about it.
I'm thinking about it. Oh, okay. So in the beginning, you were saying, I think you might have mentioned something about how you wouldn't want to apply it to time course data.
No, you can.
You can?
Yeah.
Okay.
That might even be better.
Yeah.
Okay. I didn't show this either, but I have in one of my projects, I have time course data, but they're like so there's 4 time points. There's like or we have like a stimulation, and then we have like so we have like unstimulated, and we have like 3 time points. And it's like it's basically like causing I guess like a developmental cascade to occur. So and we have like some a priori knowledge about like which time points should have which like like the cells at different like parts of the cascade. So we like subsetted our data and then did pseudotime to just like use those time points. And it like looks like it kind of lines up with what we expect.
So are there any special considerations that you have to do?
I think that was like a lot of like thinking about it like from the biology perspective in order to come up with that conclusion. But there actually are a couple of like the review at the start that I've talked about at the very start is pretty good for understanding like different types of trajectory methods. I would definitely recommend this review and the benchmarking paper as like a background. But there are a couple tools that are specifically like they only work with time series data. I think in most cases we just have the snapshot data.
So do those tools that only work with time series data do supervised pseudotime
Yep,
then?
they do
Like,
supervised.
labels for each
Like
cell?
the algorithm. I know this like I don't really know what this means, but I've heard this term a lot, which is optimal transport. But it's like an optimal transport algorithm that takes into account the like time point, assuming that you have a actual time series data. But and I was like interested in running that algorithm, but it seemed like by the time it came out like around the same time as a lot of these ones, like 2019, but they had like a massive data set. Like for back then, they had like 300,000 or 400,000 cells, which is like larger than most data sets back then. And it was not clear to me if you could run it on fewer time points. I think they had like 6 or 7 time points. So their paper was not clear about like what are the limitations. Can I do it with 2 time points? Can I do it with 3? Like can I do it with fewer cells?
What about like the spacing of time points?
That's a good question.
You
I think
know the answer to that?
yeah. I think that if you don't have like the intermediate state, you're not going to be able to run pseudotime.
Oh,
on
but
it.
do the spacings have to be the same for each?
Oh. I mean, that's like it probably doesn't have to be, but like that's a good consideration, I think. This is where like if you were smart, you would do like the experimental design, like in line with what kind of data analysis you want to do, which like never really happens. So
Also, do you think we can use this for like maybe the treatment? Like genes
like
that
transition from home like cell like regular cell to cancer cell.
yeah, because sometimes they go into like a quiescent mode that I think is like cancerous. So like,
Yeah.
over time, and then all of a sudden they go like
That's a good question, but I think like that's like I think a related field is like lineage tracing, where like like you have a cell and like it divides and then it divides again and divides and eventually it's cancer, right? So there's like an actual lineage, and I think there's some like experimental methods to keep track of cells from the like lineage perspective.
So maybe this works in neurons and not brain, but.
I think that you could apply it to cancer or something like that, but it would maybe be more appropriate to use something else.
Yeah, so I know there are like clonal evolution, like single cell DNA analysis tools. So like literally just figuring out what mutations they have and like how similar are the mutation profiles to like the ones before, and you can kind of infer a trajectory of mutation burden or accumulation over time, which is like, that's crazy to me. But yeah, I don't know. I'm sure you could do it in cancer, but like cancer, you always have to be aware of the fact that the DNA is also changing. It's not just the RNA.
What you said could also be interesting in like haploid genes, or I don't know, like mutation burden in familial Alzheimer's risk or something like that.
Yeah, I have no idea if that is a thing.
I think that like cancer is a special case where mutations are like, because like Alzheimer's, we're thinking about like somatic mutations, not like these like actively occurring mutations in like dividing cells. Unless maybe Alzheimer's is just cancer and we didn't know.
No, yet.
That's why it's been so hard to study.
Okay, any more questions? Okay, thank you all for coming. I'm gonna turn off the Zoom.